Okay. Good afternoon. Can you hear me? Okay. We would like to begin by thanking the Emblema Foundation for supporting all the national research centers presented here at the 2025 Osaka Expo and the Italian Pavilion for hosting us today. Today, along with the distinguished panel of speakers, we will share how our center is advancing medicine through innovation. We would like to begin by presenting a video that introduces our National Research Center. Thank you. My name is Emanuela Roman. I am the Research and Communication Manager of the National Center for Gene Therapy and Drugs based on RNA technology. And I would like to start by welcoming Professor Rosario Rizzuto, who is the president of our National Center. And as a professor and director of the Department of Biomedical Sciences of the University of Padua, he leads Spoke 4 of the National Center. Please, Professor Rizzuto.
Good afternoon. I'm here as president to welcome our Japanese hosts, since we are here in Japan. And I must say that I'm really very glad that we have the opportunity to put our center in contact with the Japanese colleagues and the Japanese companies. Italy and Japan, I think, have a lot in common. They have a long history of culture, arts, good food. And maybe in part because of all this, we have in common one aspect, which is longevity. And longevity, of course, is nice, especially when you advance in age, you like it. But also is a medical challenge, if I may have the first slide. Because the next. Because an aging population in health needs to address a number of medical issues. And one of the national centers that the Italian Ministry of University and Research decided to create was on medical technologies, and that you may expect. I mean, computing, mobility, biodiversity, agriculture, big themes are the other four centers. In the case of medicine, which is strategic for our country and for your country, the National Center is focused on a very narrow topic. And the very narrow topic is RNA and gene therapy. So why RNA and gene therapy? And I think we really have to look at what medicine is now. And medicine, because of our understanding of the molecular basis of rare and common diseases, has become an area in which we are capable of addressing specific patients, the molecular identity of the disease of a single patient. And this is the task. Precision medicine, individual medicine, however you want to call it, means that in the current practice, I wouldn't say in the future, we need to be able to identify first and then cure the specific molecular defects, which means that this is a great opportunity because it means having more precise, more effective, less side effect therapy for patients. But it also means that there is an intrinsic risk, or let's say it, an intrinsic cost, which is that we don't need to develop one successful drug, two successful drugs, but we need to develop hundreds of successful drugs because we need hundreds of new therapies for individual patients. And this great scientific and medical opportunity can become a big risk for the health system because it means that we need to use medicine that are developed for a small number of patients, which means medicines of high costs. And unless you develop and utilize and possess the technology for developing this large number of drugs, you will end up either deciding that you cannot give to all patients the best possible drug, and this is what we should not do. Our universal health system in Italy is a great strength. Every citizen has the right to have the best possible cure. But if we want to maintain this, which is a social, an ethical principle, then we really need to be very successful in developing the new technologies. And if you think of what are the most flexible technologies now for addressing every single drug target, well, these are the ones that are based on nucleic acids. On RNA, with RNA you can make proteins, with RNA you can silence genes, with RNA you can modulate the function of individual genes. Or gene therapy. One thinks of gene therapy as introducing a defective gene in a rare genetic disease. Gene therapy today is much more than that. Gene therapy is therapy of cancer, is therapy of common diseases. So now these two platforms, RNA and gene therapy, are really flexible platforms that we need to develop and utilize for new drugs. And if I can have the next two slides, I will go very briefly because you are going to hear the spokes. Just to mention that you will hear about the so-called vertical spokes, which are spokes on disease areas, going from disease pathogenesis, so understanding the fundamental defects in a disease, to the development of drugs. And you will hear about genetic disease, cancer, neurodegeneration, metabolic and cardiovascular, inflammatory, infectious, and the methodological spokes or horizontal spokes, the ones that are needed for every one of the groups that we discussed before. And therefore, the RNA chemistry, the biocomputing, the platform for delivering, for sending the nucleic acid to the right organ, the pharmacology for analyzing safety and efficacy, and finally, spoke 10, which will be the GMP manufacturing and the clinical phase, the very last phase of the process that is handled by a specific spoke. And with this, I stop, and we have the director.
Thank you very much, Professor. Our next speaker is the Program Manager Director of our National Center, Dr. Elena Quagliato.
Okay. Good afternoon, everybody, and I'm very proud to be here at this important event at the Expo 2025 in Osaka. It's the first time for me in Japan, and I think it's a great opportunity to be here to explore, to foster international collaboration for this national center. So we owe much appreciation to the INARC project, which allowed us to be here today, and which is a collective showcase for Italy's 5 national research centers. They were funded under the umbrella of the National Plan for Recovery and Resilience, which was the response to the pandemic crisis in 2019. And they were funded to deliver key enabling technologies in different strategic fields. So here you see on the map how they are geographically located. So we are located, for example, the RNA gene therapy is located in the city of Padova. I will just give you a short presentation, just a general framework of the National Center, and then I give the floor to our scientists who will talk about research results and research activities. So the National Center for Gene Therapy and Drugs based on RNA technology was a big project proposed by the University of Padova. It was funded on June 2022, and with an allocation of money of 320 million. So the project was launched on 1st November 22, and it has an estimated duration of 36 months. We are in the 3rd year of the project. So we are very committed with themes, the Osaka Expo themes, by saving lives, delivering personalized medicines, empowering lives by expanding international and fostering international collaborations, and connecting lives by communicating, of course, new ideas. What is the mission of the National Research Centers? Professor Rizzuto underlined it already. I just repeat that the National Center carries out research activities and promotes innovation at national and international level. And it wants to identify and validate gene therapy and RNA-based treatments, new treatments, developing and bringing new pharmaceutical products to the market, and of course taking action towards a more equitable and sustainable national healthcare system. So it's something really unique at national level, but because of its features, we think it's something, a great opportunity and something unique also at international level because of the specific fields of research, the revolutionary fields of RNA-based drugs and the gene therapy. Because of its critical mass, we have more than 1,200 researchers involved, and we recruited more than 500 PhD students and researchers during these 2 years of the project. And we have a network of 36 public and private entities. And of course, because of the allocation of money that are 320 million euros, that has to be used to fund research in these specific fields. As I said, we have a big network of 36 partners. We have public universities involved, a private university, hospitals, we have private foundations, but also you see pharmaceutical companies such as Sanofi, AstraZeneca, Chiesi, Dompé, which are Italian, but also international companies. The structures, the model that we use, it's a little bit complicated. So the National Center acts as a hub, and I work for the hub. So we manage the allocation of funding to the different spokes, and we coordinate the activities. And the hub unites 10 different spokes that aim to translate research results into the development and manufacturing of personalized medicine. So the research activities are carried out in the spokes, which group together public and private partners to work on a specific field. And here, so Professor explained it already, so we have the horizontal spokes that cover research areas of human disease, spoke 1 to spoke 5, spoke 1 to spoke 5. We have the 5 different areas of human disease. See the vertical spokes, sorry, I just inverted them, okay? And they focus on identifying the most promising candidates for RNA drugs therapies across genetic diseases, cancer, neurodegeneration, metabolic and cardiovascular diseases, inflammatory and infectious diseases. And then we have the other spoke from 6 to 10 that are involved in transforming the research results into technological products. So you have RNA drug development, biocomputing, platforms for RNA and DNA delivery, pharmacology safety, and clinical trials of gene therapy medicinal products. Our three main pillars are the technology transfer. We nominated a technical scientific committee vetting 34 projects with an allocation of 60 million euros in order to transform our research results in tangible products. And then we invest in infrastructure, so we are going to build an RNA production platforms in the south of Italy. There will be the implementation of a gene therapy center in existing labs at the Ospedale Pediatrico Bambino Gesù in Rome, San Raffaele Teleton Institute, and Tettamanti Foundation. And of course, there's in act the modernization of labs with new equipment in the different spokes. And then last but not least, we invest in human capital. We have 700 researchers involved, as I previously said. More than 500 were recruited during these 2 years, young researchers and PhD students, and 60% of whom new recruitments are female, because one of the targets of the National Plan for Recovery and Resilience was to overcome gender gap. We also support micro, small, and medium enterprises. It's crucial for economic growth, innovation, and job creation. And with around 30 million euro were assigned through cascading calls, and 60% of this budget were allocated in entities in southern regions of Italy. And what about life learning initiatives? We set up a new national PhD program, so a 3-year program in RNA therapeutics and gene therapy with more than 10 Italian universities involved, with a specialized course curriculum, and then a postgraduate program, 6-month program, which is called Pharmatech Academy, with internship for the students at companies throughout Italy. And yes, of course, so I said that these two training courses are both located in Naples, in the University Federico II of Naples, because another target of national plan was to address regional fragmentation, and this is why the 2 educational programs are based in the south, in Naples, in southern Italy. We have, I said, the hub office are located in Padova, but we also have another office in Naples, and the RNA production platforms is going to be built in Naples. Okay, so I think I'm running out of time, and these are just a few slides showing how we try to advance medicine through innovation. Just here you see, for example, the CAR-T therapy process, which is specific cancer treatment, which is used by Ospedale Pediatrico Bambino Gesù in Rome. And here the digital twin. And so I conclude just saying that our future starts now. We are really working to be at the forefront of a medical revolution, and we just have to work to allow future generations to access critical treatments. And many thanks. Here you find our QR code, so you can go to visit our website with detailed information about the center and all events we are involved. And you find also our social media channels, LinkedIn and YouTube, that we use to engage with public. And I would like to thank you for your attention. I hope I evoked your curiosity about the National Research Center for RNA and Gene Therapy. And if you are interested in collaborating, partnering, just learning something more, please contact us. Here you find our contact details. Thank you very much for your kind attention.
Thank you, Elena. Now that you have a better understanding of how the National Center is organized. Speakers from each of the spokes will offer deeper insight into how we are advancing medicine through innovation. And we begin with Spoke 1, represented here by Professor Alessandra Renieri, who is a professor of medical genetics at the University of Siena and director of the medical genetics department at the Siena University Hospital.
Thank you so much. I'm very glad to be here, and thank you for hosting this wonderful city, Osaka. Yes, in addition to my role at the University of Siena, I have also a commitment at the European Medical Agency, and you see me here in the new building in Amsterdam. Every month I go there for the CAT committee, for the Committee for Advanced Therapies, for the authorization of the final product of a project like this or similar that are gene therapy, cellular therapy, and regenerative tissue engineering. As a geneticist, I am so much involved in this because this is a new era in which genetics is switching from the diagnostic to therapy, and as a clinician for helping families, as a researcher, and you will see my own project in the last slide. I'm representing the entire spoke here, and you can see the region of Italy involved: Milano, Padova, Bologna, Siena, and Cagliari, Naples, and Bari. They are spread, as you can see here, over all Italy. And I can give just a few examples during the presentation, but I have the brochure here and some copies. You can find some copies there in order to see every project. There are more than 8,000 genetic diseases, so you can imagine that each project is different, even if some of them can share strategies. I divide my presentation in the four areas of the gene and RNA therapies, and the first one is ex vivo gene addition or gene replacement. I can give you this example from the University of Modena and Reggio Emilia concerning one of the rare skin disorder that is recessive dystrophic epidermolysis bullosa. And here you have the strategy using a retroviral vector, so the Singama retroviral vector will insert the gene in ex vivo, so in vitro, and so transforming the skin, and then you can replace the skin to patients. And now you can see this project is in the phase 1, phase 2 clinical trial. As an example of ex vivo genome editing, we have here this very interesting strategy from the University of Padova. Indeed, this is a combination of genome editing with targeted gene replacement, and is presented for any neuropathic storage diseases. So this means, for example, metachromatic leukodystrophy or X-linked adrenoleukodystrophy or similar. And the strategy is to use a CRISPR-Cas9 tool in order to target the CX3CR1 gene, which favor the myeloid engraftment in central nervous system. And beside to target and so to disrupt this gene, they replace the core gene missing in the different neuropathic storage disease disorders. So these are very interesting strategies that is also patented. Concerning the example of RNA drug therapies, I can show you here the strategy. This is a bit little. The strategy of the colleague at the TGEM, Brunella Franco, this is an AAV-based delivery of an inhibitor of mRNA 181AB to prevent neuronal degeneration and treat any optic neuropathy and inherited retinal disorders which are associated with either primary or secondary mitochondrial dysfunction. So also in this case, we are talking about rare disorders, but one common strategy is in order to cure many of them. They are also working on Leber hereditary optic neuropathy and Lee syndrome, but as you can see on the right of the slide, they are also considering glaucoma and others. And you can see there also the contact for this very interesting project. Another mRNA therapy is the idea to deliver epsidin in the patients who are affected by juvenile hemochromatosis. This from Petrangelo from University of Modena and Reggio Emilia, who is also the SPOC1 leader. Then we can go to the in vivo gene addition. The past, the word was gene replacement, but this is not the proper word right now. So we have here the interesting strategy to deliver a peptide, and this is a PEDF-derived peptide in the AAV2 vector with the in vitro injection for any inherited retinal degeneration. So also in this case, the target is not just one rare disorders, but more rare disorders that have in common retinal degeneration. And you can see here also this strategy is patented, so they are ready for company who want to develop further. So here we have also another strategies from TGEM, and this is in vivo gene therapy for Marotolami syndrome patients. You see patients there with infusion of AVV28, TBG, ARSB, so the gene disrupted. And you can see here that high dose is very much effective, and now we have 5 years post-gene therapy follow-up, and so this is very, very, very promising strategy. Then we go to the in vivo genome editing. So the most challenging, but potentially the most effective strategies. And I can present here 2 projects from our university, both patented. One is for Alport syndrome, a disorder that involves kidney mainly, but also hearing loss and additional eye problems are present. And here we have a dual CRISPR-Cas9 system delivered by AAV9, and we demonstrated in the in vivo and the preliminary in vitro system, mainly consisting in podocytes derived from the urine of patients, that we could have a correction up to 58% of one of the hotspot mutation of this disorder. And now we have also a humanized mouse model. Humanized mouse model means that you can use exactly the same drug that you have used in the cellular system, and so this is a warranty for working. And the second project is a neurodevelopmental disorder, so a very famous neurodevelopmental disorder that is the second after Down syndrome in girls, which is Rett syndrome. And in this project, we are targeting the 4 main hotspot mutations, covering more than 5% of patients. By this project, we delivered this dual CRISPR-Cas9 system also in the AAV9. Here we have also organoids preliminary data, and then, of course, the mouse model. So also this is ready for do something at the level of clinical trial. And the last slide is dedicated to one of the problems that we face with talking about genome editing, that is the off-target analysis. So nowadays, it's very difficult to identify companies or strategies or protocols in the literature covering this problem that is mandatory when you want to develop a drug based on genome editing. So we took advantage by the presence in Navio University, one of the most advanced technology for sequencing, the long-reads sequencing platform of Oxford Nanopore, and we developed a specific protocol for covering this very big issue, the identification of target. Thank you so much for your attention. We are looking for talking with companies that are interested of any of these projects. Thank you.
Thank you. Now we welcome our next speaker, Professor Elisabetta Ferretti, who is Professor of Pathology at the Sapienza University of Rome, representing SPOC2.
Hello, everyone. I'm very excited to be here and I'm honored to represent the SPOC2 cancer of the RNA Gene Therapy National Research Center. The title of my presentation is Using Advanced Technology, specifically spatial OpenST, to identify RNA biomarkers and find novel RNA-based treatments in cancers, in tumors. SPOC2 cancer pillars is focused on the development of RNA-based therapies to fight cancer and their metastases. And our work is organized around three main pillars. The first one is to identify RNA targets and biomarkers in human cancer. The second is to explore how therapeutics can help boost the immune response against cancer, increase tumor immunogenicity. And the third is develop RNA-based anti-cancer therapies, including vaccines. So in the first pillar, a group of us focus their efforts on the develop of new spatial omics technologies. And now I will explain you why. In our first generation of transcriptomics, analyzing tumors mix all the RNA together and like blending cells into a smoothie. So we lose important spatial information. And even if with single-cell omics, we get RNA of each cell, but still don't know where each cell was in tissue. But in cancer, location really matters, especially to understand how cell interacts. So that's why spatial omics are a big step forward, and they let us see both gene activity and where it happens without destroying the tissue. So that's why in 2020, spatial transcriptomics has been declared by Nature Methods the innovative technology of the year. So you can think about, we have now a sort of microscope that show not just colors, but all RNA at once in a sample. So spatial transcriptomics help us understand the molecular mechanism behind cancer. However, when we start the study, need methods that are easy to use, with high resolution, cost-effective, and can be scaled in 3D. That's why a group of us at Sapienza University, in collaboration with the international collaboration with Berlin and DC, developed OpenST, a sequence-based tool to address these challenges.
And indeed, with this type of platform, we can starting from a single tumor samples, as you can see here, and capture a wide range of RNA in a biased way, and map cell identity in millions in both cancer cell and stromal cells, then define cell biomarkers, identify rare cells with lowly expressed RNA, capture many genes per cells with single cell, and molecular resolution up to subcellular level, and gain insight to cell-cell interaction. And what is another important point of these technologies is that it is highly versatile and easy to implement. Indeed, not just mRNA, as other technologies already commercial. We are now working to capture all types of RNA, including non-coding RNA. And it is much more cost-effective compared to the other approach. So we, in our first set of experiments, have just analyzed mouse brain, head and neck tumors, and lymph nodes in both 2D and 3D. And we were able to identify different features in immune strong tumor cells. And we can say that with 3D analysis, we can see more than in 2D analysis. And within this pillar, we also spend effort on the novel diagnostic markers in terms of RNA, circulating RNA. Particularly, we focus our attention on prostate cancer, that is one of the most common cancers, that now diagnosis is based on mRNA and PSA levels. And we proposed a study to test diagnosis shift that sees circulating RNA and mRNA biomarkers as the pre-biopsy triage for patients at risk. And with this first study, when we have a first cohort for discovery and the second cohort for validation, we identify mRNA biomarkers that together with 2 microRNA are clinical significant cases with a good score for tumor aggressiveness prediction. And this is now in a first clinical trial for further validation. In the second pillar, we have in the field of identification of strategies to increase the tumor immunogenicity, group from Siena, coordinated by Professor Michele Maio, developed a platform that produces a cancer vaccine using an epigenetic approach. As you can see here, the process starts with collecting immune cells from patients and continues through several steps, including demethylation, this is a fundamental step, to activate enhanced immunocells and before reinfusion in patients. So these strategies have advantages that are multivalent strategies, meaning that it can trigger a broad immune reaction, and it has high immunogenicity potential. The vaccine includes the expression of different methylation-regulated antigens. And in the at University of Padua, group coordinated by Professor Piccolo in the same context, developed an EVAX platform, creates immunoactivating cells that mimic the patient tumor, helping the immune system recognize and attack cancer more effectively. And as far as the third pillar, we have some studies from University of Milan, coordinated by Professor Mantovani. They first identified a sequence of RNA that are abnormally present in breast cancer and stomach cancer. So they decide to develop antisense oligonucleotide, chemical modified, that are designed to block exactly and only abnormal RNA form. And they've already used successful performing therapeutic modulation of this splicing RNA. And in this context, in University of Milan, coordinated by Professor Minucci, research group dedicated effort to sequence optimization for a better antigen processing and presentation, since existing cancer mRNA vaccines exhibit limited therapeutic efficacy. Thus, this group worked out on mRNA platform for the modification of the expression of specific MAH molecules and antigen-presenting cells. Within third pillar, we have also a group from University of Catania that's worked out on peptide coding mRNA that inhibits mitochondrial activity in cancer cell and is able to produce a pro-apoptotic effect. And the efficacy and pharmacokinetics of such peptide delivered in virus or nanoparticles are going to be surveyed in a model. And at University of Sapienza in Rome, a group coordinated by Professor Boffi worked on new carrier for delivery of RNA-based therapeutics, and this is very interesting in a target manner, using a CD71 reporter that is expressed mainly by cancer cell. And this is subject matter in object in Italian patent application. This is the last one. At University of Turin, a group coordinated by Professor Taverna showed that microRNA delivered by an aptamer is able to impair metastasis formation in two types of cancer, melanoma and breast cancer, and the research applied for a patent for chimeric aptamer discovery. So I conclude that target biomarkers discovery with new technologies to innovative delivery system, research involved with SPOC2 are shaping the future of cancer precision medicine. For any other information, please contact the email that I reported here, and I thank you all of you for attention.
Thank you very much. Please welcome Professor Gian Gaetano Tartaglia, who is a principal investigator at the Italian Institute of Technology and coordinator of the RNA flagship, representing here SPOC3.
Thank you very much, and I'm very happy also to be here with you. And I'll give you a little bit of a presentation about what happens in the first SPOC, mainly related to neurodegeneration. All right, so especially today, I'll be discussing the designing and engineering of RNA molecules with the purposes of changing the expression of genes or inhibiting or altering the function of proteins. And I'll give you a couple of examples, quite detailed, and I'll give you also some other names that are being dealt within the context of our PNRR projects. So the first example relates to synapse. That is a class of RNA molecules that are being discovered by Stefano Gostincic in our institute. And these molecules are particularly amenable for engineering. Indeed, what do they have? They have two domains. One that is binding to a piece of RNA, most often in the 5' UTR. Another one that is called the effector domain that recruits ribosomal components. So differently from other technologies, such as, for instance, ASO that you might have heard, or anyway, inhibition through RNA, SIRNA, this class of molecular synapse allow to potentiate the translation of a gene, and therefore are particularly relevant for cases such as, for instance, related to haploinsufficiencies. And this is just a sketch to show how they work. So you need to know which strand is involved in the transcription. And you have a piece that is binding to that part of the RNA, and another element that is the effector domain where the ribosomal components are attached. By having this effect of attachment of ribosomal components, the translation of the gene increases up to 2 to 5 folds. So this is particularly relevant, as I was saying, in cases such as, for instance, FMR1. There is a gene that is, for instance, only expressed on the X chromosome. So in the case of there is a particular problem in this kind of haploinsufficiency, the synapses are particularly useful. And this is quite important, the engineering of these molecules, because you can engineer both the region where the binding occurs, so therefore for every RNA that is transcribed, and also you can potentiate or depotentiate the binding of ribosomal elements, so by introducing IRES and other elements that are particularly important for recruiting RNA. Right, so this is one of the classes that we have used. The other class, already introduced by our colleague, that is called aptamers, relate to the designing from scratch of RNA molecules that, in this case, bind to proteins. In our spoke, which is related to neurodegeneration, as I gave you earlier, we're particularly interested in cases related to ALS, so amyotrophic lateral sclerosis, as well as Parkinson, and there are also other cases such as, for instance, the Kabuki syndrome. But this is just examples that we have considered as the main ones. And with my group, was involved in some of the biggest projects related to TP43 and FAS that have actually led to a couple of interesting patents in the field. I also like to mention that the technology is so powerful that also can be applicable to other cases. Indeed, there is a case related to RAD51, so the other spoke 2 that we have just heard about. So the novelty in our own project led to the fact that we have developed a technology to design these aptamers that is quite different from what is historically known in the field, that is the SILEX. In this particular case, we use a completely in silico pipeline that allows us to control for the affinity as well as the specificity of the RNA towards the target. Of course, you can use this technology for many purposes, and you can use also different algorithms or different procedures. We use one that is called CatRabbit, but mainly if you just want to break it into the two classes, diagnostics versus therapeutics, we can use these molecules to visualize where the protein is in the cell and also in the tissue, as I'm going to give you in a moment, but you also can use it to interfere with the function of the proteins, so therefore you can have a therapeutic effect. All right, so let me give you an example related to ALS. And so this is a beautiful image from a recent paper in Acta Neuropathologica. So what you have here, this is ALS, this is a TDP43, a protein that aggregates, all right, in neurons, in the motor neurons. And in this particular case, the aptamer is used for staining. So what you see here, where is this arrow pointing to the neuron, is the visualization of the aggregate thanks to the aptamer that is binding to the aggregate specifically. And this has allowed to achieve a sort of resolution that you can't otherwise through antibodies. And this is a more detailed case that I'm going to give you. This is in vitro instead. What you have here is the comparison between what you obtain using the aptamer, which is on the left lane, and what you obtain using the golden standard in literature so far, which is the thioflavin T. As you might see, this is a case where the aggregate grows in time, and the aptamer is capable of showing step by step the growth of the aggregate, while the thioflavin T, which is a pretty used dye, can't. This is because the molecule is particularly small, also intercalates well with the aggregates, and allows to detect even 20 nanometers in size aggregates. All right, so this has led also to applications in diagnostics. These are brain homogenates, and comparison with what you obtain using the classic antibody separation into classes indicates that the aptamer can really differentiate across stages of the ALS. So this is being patented, actually is becoming quite an important element in the diagnostics. But of course, diagnostics doesn't mean much if you don't do therapy, in my opinion, at the same time. So these aptamers actually are quite interesting, collocated in the area of diagnostics. So what you see here in this plot is just the potential of RNA, which is, in my opinion, when Professor Rizzuto was speaking before, I was thinking about this slide, because this slide shows you that these two different RNAs can do completely different effects. Here we are talking about the aggregation of the protein TDP43, and there are some atomers that completely block the aggregation. On the left, I'm sorry, the slide is detailed, but take my word, then I can tell you the biochemistry behind. On the right, instead, you take another piece of RNA, or you engineer, as in our case, another piece of RNA that instead promotes the aggregation. So with the same molecule, depending on the structure, the composition, you can basically polarize the effect towards aggregation or the opposition. Right, so if you use the beneficial aptamer, the one that blocks aggregation in the cell, you do observe actually the effect that you have wished for. So in this image, you see that small dots that correspond to the aggregates indeed are dissolved in the presence of the aptamer. So we have found, in a way, and also patented, a molecule that has a great potential for therapeutics. Similarly, and this is why I'm going to go quite fast, this has been done for other proteins, such as, for instance, FAS, that covers another part of ALS, also frontotemporal dementia. And this data here just showing the biochemistry, and also that the molecule can be used for solubilizing and also you can apply the same principle to RAD51, that is a protein involved in DNA repair. In this case, the aptamer indeed blocks the repair, which has an important consequence for the metabolism, especially in cancer cells. And so this also has led to a patent, and it is a very specific case, the one I'm showing here. There are many other aptamers that have been developed and shared in the community, and related to, for instance, ML4, HUD, and other proteins that are neurodegenerative disease related. And so these are the people who have been doing the designing and the experiments. Thank you very much.
Thank you, Professor. Oh, great. Next, we welcome Professor Rosario Rizzuto again, our president, but also coordinator of Spoke 4.
So I change hat now. I move to the hat of the scientist. And I will show you some examples of what is being done in the Spoke on metabolic and cardiovascular disease. The Spoke on metabolic and cardiovascular disease is divided in work packages. First of all, you look at where the groups are located, and you see that in the goal of the research center or the national center to unite Italy in this new effort. We have universities and research centers from the north, Milano, Padova, Trieste, and from the south, Naples, Bari, Catanzaro, and Palermo. The topics are three. So metabolic and cardiovascular, I suppose that you expect two of them for sure. One is Work Package 2, which is the deadly triad of diabetes, obesity, and hypertension, what our endocrinologists would call the metabolic syndrome, and the Work Package 3, which are heart diseases. To these, we add one Work Package 1, which is an orphan area, which is skeletal muscle, not only because it is in many aspects close to the other striated muscle, which is heart, but also because muscle metabolism, muscle atrophy, and aging have an important part in age-related disease. And I will start with Work Package 1. And Work Package 1 addresses sarcopenia. Sarcopenia is the loss of skeletal muscle force that we have in aging, but also in a number of chronic diseases. From cancer that we heard about to metabolic diseases, to kidney disease, and so you lose skeletal muscle. And losing skeletal muscle is a precipitating component of the disease itself, because being weaker means not going out, and going out being immotile, and being immotile means venous thrombosis and osteoporosis and so forth. So it's really an important target, and it's a target for which we have no real drug. We don't have good drugs addressing skeletal muscle loss. So one of the groups, and this I will focus on One which is the work of Marco Sandri in the University of Padua. He's a world expert on the pathways controlling muscle wasting, so the autophagy and proteasome-related degradation of skeletal muscle. And in particular, he identified and studied in detail MRF1 as a skeletal muscle component regulator of the ubiquitin proteasome system, and FOX1 and 3. And what he did was try to address one condition of muscle wasting, which is cancer-related muscle wasting. You see cancer-related cachexia. And in this model, he utilized shRNA, so an siRNA-based approach for silencing the atrophic pathway. And he did this by electroporation or direct injection of an AAV. And as you can see, and after he collected the muscles and looked at the muscle mass, and you see that when you silence FOXO1 and 3, or when you silence BRF1, you have a recovery of muscle mass in the cachectic, the cancer animal. And what is striking is that if you make a combinatorial treatment with the two siRNAs, you have almost complete recovery of muscle strength. So this, as far as cachexia and sarcopenia is related. Work package 2 is a massive work package, as you can see, because you have metabolic syndrome, you have hypertension, you have diabetes, and a number of individual targets have been identified, proteins that do have a role, and you see ranging from P66-SHIC to the regulators of some microRNAs regulating kidney dysfunction, to again P66-SHIC in the ischemic wound, and in atherosclerosis, a pathway of cholesterol control, and in hypertension, and some microRNAs, and in fatty liver, regulators of fatty liver. In obesity, I will, again, there are a number of potential targets. I will focus on two. I will present you very rapidly a couple, which will be our own for simplicity, which is mitochondria. You asked me about mitochondria, so you will see mitochondria now, and how mitochondria are related to the inflammatory obesity, and you will hear something about atherosclerosis. And why mitochondria? Because we want to think out of the box, and think out of the box means not identifying the most obvious target, and the most obvious target, of course, in inflammation would be some pathway for synthesizing metabolic mediators of inflammation. Well, we know now that mitochondria do control the inflammasome, therefore the production of the inflammatory mediators, and in particular the calcium signals, not only calcium signals, but calcium signals are regulator of mitochondrial oxidation, mitochondrial DNA oxidation, and therefore pro-inflammatory component. So what we have done is to inhibit calcium fluxes in mitochondria, and you can do this by silencing the channel that brings calcium in the mitochondria. In the middle graph, you see that if you do this in vitro, you have a drastic reduction in inflammatory mediator production. And what is particularly interesting is that if you do the experiment in vivo, here we have a model of inflammation, which is a simple model of inflammation, which is joint arthritis, gout, that maybe many people know. And if you inject an siRNA to the mitochondrial target, you have a drastic reduction in the inflammatory process. So we have a totally unexpected new target in inflammation, which is a mitochondrial protein. For atherosclerosis, we refer here to the regulator of the cholesterol biosynthesis. And again, you see that by injecting the this regulator, you have a down regulation of the atherosclerotic condition. As far as heart is involved, I present you very rapidly 3 different projects. One is Mauro Giacca in Trieste, and he has been doing very elegant studies on identifying cardiac protective proteins produced by the liver. So he screened the number of secreted proteins through functional selection in isolated cardiomyocytes. And then when he identified new targets, chordin, and when he made an RNA drugs, an mRNA for chordin administered in myocardial infarction, he sees that he has a significant recovery of muscle mass, which is very, very interesting, and I will not spend time for the others, which would be cardiac regeneration after myocardial infarction. Also, in this case, he identified some positive regulator of cardiac regeneration, and he did some editing also. And he has some microRNAs that are beneficial, that can be targeted in cardiomyopathies. The group in Milan of Humanitas, what they have been doing is they identified cardiac-specific postnatal long non-coding RNAs, which is protective for heart failure. So the group of Gianluigi Condorelli, what he did, they identified these long-coding RNAs, and again, when administered in vivo to models of heart dysfunction, they are protective. They made CRISPR-Cas base editing of genetic cardiomyopathies, and then they are developing an immunotherapy, again, to heart failure by identifying autoantigens that are upregulated in this cardiac dysfunction. And the last example that I will show you are some preclinical studies by the group of Silvia Priori in Pavia. And she has two models. One is a genetic form of ventricular tachycardia, which is due to a mutation of RNA receptor type 2, so the calcium channel in the sarcoplasmic reticulum of the heart. And when she silences the mutant type 2 RNA receptor, she has a normalization of the genetic model in mice. And even further, in the analysis of animal model of a long QT syndrome, again, genetic cardiomyopathies. This is in the pig, which, as you know, is the best model for cardiovascular diseases. The mice have a spectacular heart. They're very, very difficult to get them to have a cardiomyopathy. And when she made an AAV-mediated allele-specific silencing of the mutated protein, she has the recovery of the cardiac syndrome, both electrically and clinically. And with this, I'm done, and I thank you. I apologize with the colleagues that I could not mention, but otherwise I could have given no detail at all. But if you want to hear something on the three areas of the spoke, just write to me or write to Cristina Mamukari, who's much better than me with emails.
Thank you, Professor. Now, last but not least, please welcome Professor Francesco Dotta, who is a professor of endocrinology at the University of Siena and coordinator of Spoke 5.
Good afternoon. So many thanks for being here to listen to me, and for me it's a privilege to show you the activities within Spoke 5, which is devoted to infectious and inflammatory diseases. So let me start with introducing the components of the network of participating to Spoke 5. So we have University of Milan, Humanitas University, University of Padua, of Verona, of Florence, Siena, Roma Tor Vergata, and Naples Federico II. Together, this network is pursuing those specific goals. First, to discover novel RNA-based therapeutic targets in autoimmune and inflammatory diseases. Second, to engineer intelligent nanovesicles delivering RNA-based drug to treat autoimmune inflammatory diseases. To develop innovative vaccines and prophylactic drugs against novel emerging viral pathogens. To generate vaccines and/or neutralizing monoclonal antibodies against antimicrobial resistant bacteria. And last but not least, to identify disease-associated RNA biomarkers. To pursue the above-mentioned goals, we have divided our spoke into 4 different work packages. The first one, which is coordinated by University of Florence, is involved in novel mRNA vaccines, prophylactic drugs design, and immune response profiling. The second, coordinated by University of Rome Tor Vergata, deals with innovative strategies to counteract novel antimicrobial resistant bacteria and viral infection. The third one, which is coordinated by myself, is involved in studying circulating RNA therapeutic target discovery and delivery strategies in autoimmune inflammatory diseases. And the last one, Work Package 4, coordinated by University of Verona, is involved in studies on RNA-based drug design. Taking into account the time available, I took the liberty to choose a couple of work packages to show some ongoing activities and results. Starting from the innovative strategies to counteract novel antimicrobial resistant and viral infections, let me mention the work performed by the group of Giuseppe Novelli in Rome, Tor Vergata. Basically, what Giuseppe and his group are doing, they are studying the COVID-infected cells, in particular coming from the lungs, and what they found is that infected versus non-infected cells express differential microRNA profiles. And so he wants to base the approach on these differential microRNAs to generate target gene circuit that specifically activate Cas13 in order to knock out viral COVID genome in infected cells, then to formulate RNA-targeted nanoparticles for delivery into cells, and finally, in the last step, to aerosolize nanoparticle DNA complex. And basically, this is what the basis they are doing. This gene therapy construct that uses microRNA to target expression of a specific therapeutic gene only to infected cells. Basically, what they do, they have a GFP is used to demonstrate a proper functioning circuit that activates only within infected cells. And these cells that express GFP are shown in green. Then they target a vector with an additional marker gene, which is RP, that glows red. And in this case, all cells, both infected and non-infected cells, are stained in green, in red. And that's the scenario that basically you have. Then you have to go the gene circuit, all the cells are red, but only the infected cells are green when the circuit is activated. Now switching to the second work package, which is on circulating RNAs as potential biomarker of disease, I will present our work in a specific form of diabetes, which is type 1 diabetes or insulin-dependent diabetes, which is indeed an autoimmune disease. Basically, we know very well that being an autoimmune disease, the insulin-producing beta cells are destroyed by the immune system. But the recent findings in this disease discovered an heterogeneity among patients. In other words, the immune cells that are involved in beta cell destruction differ from patient to patient. So considering that we are now facing a kind of paradigm shift in type 1 diabetes treatment, because so far we only had insulin. And with insulin, you treat the disease, but you do not cure the disease. Now we have starting immunomodulators in which you may start to cure the disease in set of patients. So that's why you need biomarker to uncover disease heterogeneity. And as a candidate biomarker, we concentrated our attention on a class of non-coding RNAs, namely microRNAs, that in addition to be very important in regulating gene expression in cell populations, they are excellent biomarkers because they are secreted into human biofluids. And not only this, but it has been shown that beta cells producing insulin and immune cells, they have a dialogue mediated by microRNA. And microRNA from the lymphocytes can damage the beta cells. So that's why we are interested in studying the circulating microRNA. And what we found is basically we did a work in taking the advantage of a collaboration within a European consortium. We took hundreds of patients with type 1 diabetes at diagnosis, and we followed them up for 2 years. And at visit 1, so at diagnosis, we screen these patients for microRNA profiling. And to make a long story short, we identify 2 clusters of patients that you can see is called cluster A and cluster B. that can be very easily distinguished by simply measuring at the end 3 microRNAs. And these two clusters of patients are very different in terms of insulin need, rate of beta cell destruction, and importantly, the involvement of specific T cell subsets. The good thing is that now we have drugs that can target those cells. And so by measuring the microRNAs, we can identify in advance which are the best patients that can be targeted by the new drugs in an optic of precision medicine. The other thing that we like to do with microRNA is the role that they have in extracellular vesicles. As a matter of fact, a few years ago, in collaboration with University of Lausanne, we demonstrated that there are 3 microRNAs, namely MIR142, 3P, 5P, and MIR155, that are released by CD8 T cells and specifically goes into the beta cells and kill the beta cells. In an animal model, using a sponge against these 3 microRNAs, we have been able to protect the mice from development of type 1 diabetes and cure the disease. So we want to transfer this type of information in the human setting, and we started to use both human islets as well as human beta cell lines, in which we showed indeed that in type 1 diabetes, there are differential microRNA expression in extracellular vesicles. And the last part of this, and this is basically the last result slide that I want to show to you, is that we want to engineer extracellular vesicles containing specific microRNAs, adding to the extracellular vesicles a sponge, a Mirna sponge, so an antisense microRNA. But then the challenge is to find a strategy to deliver those extracellular vesicles specifically to beta cells. And so we want to coat these extracellular vesicles with specific aptamers that can bind only to beta cells in order to deliver the drug-based microRNA exactly to the beta cells, which is the target organ in type 1 diabetes. So to conclude, in summary, SPOC5 is at forefront of research encompassing both coding and non-coding RNAs, as I showed you, as therapeutic targets, vectors, or biomarkers for infectious and inflammatory autoimmune diseases. Overall, we know a lot on RNA and non-coding RNA. We know the function. In some cases, we can predict the functions, but still there are a lot of unknown function and a lot of discovery, and this is what we are doing in our SPOC and in our RNA center. Thank you for your attention.
Thank you very much. Perfect. At this point, we have time for questions from the audience to the first panel of speakers. So please raise your hand. I think we have someone from the staff helping with the microphone, right? So anyone wishing to ask a question or share their thoughts? If there are no questions right now, we may still have the chance at the end. Please introduce yourself first, and also let us know if you have a question for a specific speaker or to all who may want to respond.
Okay, thank you. My name is Roberto Coppo from Kyoto University. I have a general question. First of all, thank you for your very interesting talk. And my question is related to the aptamers technology. I was wondering what kind of receptor do they bind to specific target a kind of cell? I mean, how this aptamer is specific to the specific cell, which receptor?
You have a microphone on the table. Thank you very much.
Okay, thank you very much. This is an important question. In in the field of diabetes, of course, we need an aptamer that is specific to bind the surface of the beta cell. And that has been a major challenge for us, because based on the knowledge of what the beta cell express on its surface, there were apparently no receptor specific for the beta cell. For example, at the beginning, we thought about the GLP-1 receptor, for example. The problem is that GLP-1 is also expressed by other cells. So in the end, fortunately, last year was published in Nature Communication a paper from a group of scientists in the US, where they published an aptamer which was specific for a small protein on the, I think it was on my slide, and specific for the beta cell. So we contacted these people, and also in collaboration with the University of Milan and University of Padua with Giovanna Brusatin, as well as with the University of Pisa, which is in Spoke 8. We are trying to develop the appropriate strategy and possibly to find additional aptamers that are really specific for human beta cells. Anyway, thank you for the question.
Perhaps I can help you. No, I mean, basically, I mean, you, of course, can engineer a piece of RNA towards whatever target you like. I mean, if the target is amenable for interaction due to, you know, the structure and the electrostatics, for instance. So basically, the important is to find the appropriate receptor to target to. And also, for the receptor, which of the regions you can have more than one site where you can bind to. And this is also very important because you can also, you know, modulate, if you wanted, the activity of the receptor in a wish. You can switch it off completely or instead decreasing a little bit. Thank you.
Thank you. We don't have...
Thank you very much for your talks. I have a question to Professor Rosario. So in your project, there is...
Excuse me, can you please introduce yourself?
I'm terribly sorry. I'm Ryo Watabe of Nippon Shokubai. A question about, regarding siRNA delivery. So in one of your projects, there is a siRNA which targets mitochondria. So could you tell me how to target mitochondria?
Well, the siRNA that is targeting mitochondrial function is a normal siRNA to a nuclear gene. So the target for mitochondria is the calcium transporter of mitochondria that is a nuclear encoder. So it's exactly the same as targeting any other genes in the cell. So in this respect, we are not we are not introducing an siRNA or to an intramitochondrial gene transcription, but to regular. The point is, going to your question, is mitochondrial targets, I think, can be exploited in a number of disorders, in a number of degenerative disorders. But the critical point would be directing the siRNA to the appropriate tissue. This is why we went on inflammation, because the two cell types that now are known to take up siRNAs quite nicely are the liver is the king. The liver, if you want to target something which is expressed in the liver, you have no problems. You take any lipid nanoparticle, it will go to the liver. And the macrophages that, of course, take it up, but also now we know that it translocates to the cytoplasm, and therefore it does silence accurately. So if you're dealing with inflammatory diseases or liver diseases, or as you have seen, Mauro Giacca has identified a liver-produced protein that interacts on the function that it wants, which then you're fine. If you want to direct an siRNA to a different organ, say that you see a mitochondrial role in neurodegenerative disorders, for example, There, I think the main obstacle is sending the siRNA to the cell of interest, so to the neuron, and this is where I think, and I hope that the center will allow big advancement. This is why I think that it is very important to have the so-called horizontal spokes that will help us to have delivery methods that are more and more effective and cell type specific.
Thank you. So thanks for the stimulating input. Let's now move on to the second half of the presentations. So please, yes, please welcome on stage, welcome on the stage Professor Viero, Professor Elmer Citterich, Professor Brini, and Dr. Silvestri. Great. So please welcome Professor Gabriella Viero, who is responsible for the Institute of Biophysics at the National Research Council of Italy and principal investigator in Spoke 6.
So, minasan konnichiwa, and thank you for being here. I'm really honored to have the opportunity today to take your hand and work through our activities and innovations that we have been developing in Spoke 6, which is about RNA drug development. So as you can see from this slide, we have a core business, which is also our mission, and which is to think and develop RNA-based solutions for health. At this stage, I will not go into the same technical detail my colleagues went in, because basically for IP protection. But I will try to walk you through 3 main points: so research, strategic development, and also some concrete outputs. So I would like to start letting you know who we are, because I think it's important. And so I think it's very relevant for us to know that we are really top-notch scientists, a group of top-notch scientists in Italy. And we are a group that spans different disciplines. So we are not just interdisciplinary, but we are really transdisciplinary. And as you can see, we have skills spanning from RNA biology, cell biology, molecular biology, but we also have in our groups chemists and physicists as well, and you will see throughout the presentation the reason as to why this is so. So the second point that I would like to make is where we are positioned, both geographically, but also from a research developmental point of view. So from the point of view of geography, as you can see, and you've seen also from other spokes, we are spanning the entire Italian territory, which we think it's really a plus. And our spoke combines principal investigators, which are both senior but also junior principal investigators from multiple universities, research centers, and also some other institutions. So in terms of research positioning, our activities, as you can see from this slide, are primarily positioned between research and development and preclinical trials, preclinical studies, and the corresponding technology readiness levels are between 1 and 5, 6. There are a few exceptions here with higher levels of readiness, but the large majorities are in this position. And so what we do is basically trying to identify RNA target, to identify RNA compounds, but also to work on these, to leverage on these compounds and to make chemical modifications and also to innovate in terms of delivery systems. So in practical, what are we doing? We are dividing our activities in 3 main work packages that are divided functionally in the development of technological platform. I'll tell you a couple of things about that later on. Then we also have this development in terms of chemical modifications in order to increase the above-mentioned selectivity and also lifespan of the RNA molecules. And then we are also testing some case studies using our work package 3 with molecular and cell biology. So in terms of methodological approaches, they are, of course, highly transdisciplinary, as I told you. And so we are using imaging, omics, sequencing, single molecule sequencing, single molecule imaging, but also AI and computational biology. What are we doing in practice? So in practice, this is a key slide for me because this is the conceptual framework behind the entire Spoke 6. So the idea is to exploit what we have in nature, which means the cellular ecosystem, because the cell is not just producing mRNA, so messenger RNA and microRNAs, you've heard a lot about that, but also many other RNA. And basically, this is a sort of hidden layer that is not used much so far. And so our idea is to use circular RNA, the human telomerase RNA, SNO RNA, uRNA, transfer RNAs, ribosomal RNA, and also their AP transcriptional modifications, because this RNA can also be modified in order to, and decorated chemically, in order to include an additional layer of control. What's important here is that in our concept, we are using this variability, this ecosystem, in order to do 2 things. First, finding new drug targets, and the second is to use these as targets themselves. So the idea behind this is then to use these in order to expand on the existing RNA therapies that you see listed here, which are primarily using antisense oligonucleotides, so all small interfering RNA or RNA aptamers and mRNA. And our ambition is to expand on these drugs, but also expand on different RNA targets and expand on different types of molecules we are using in order to develop new RNA therapies. So some case studies. So here you see that we used some disease cases in order to probe the target identification and also the type of drugs that we are using. So you see here we have 5 cases for cancer, from medulloblastoma, leukemia, lung, breast, and bladder, and also angiogenesis, which is a more general process in cancer. But we also are focusing our attention on a couple of case studies about aging and also neurodevelopmental diseases and neurodegenerative diseases, such as ALS, SMA, and other aging. So what we are doing is that to each of these diseases is coupled one of these classes of RNA. And this, I think, is the powerful of this concept behind our Spoke. And these are the people that are involved, and as you can see, it's really a network that is well organized in order to try and answer the very same questions. So in order to develop new strategies in terms of not targets, but RNA strategies, we are primarily working on these. So, of course, ASO, messenger RNA, microRNA, but we have also other more advanced therapies like ribotech, but also RNA editing, which is really changing the sequence in situ, and also conjugated aptamers, where you can see that the RNA is conjugated directly with peptides. So also in this case, we have multiple institutions and principal investigators that are all helping in this very important network. Finally, we are also trying to generate what we call intelligent scaffold that are key because we need to deliver these molecules to the right place. And so we are also developing new approaches which leverage on bacteria, for example, or nanoferritin, nanoparticles, albumin nanoparticles, hydrogels, and others. And again, also in this case, the effort is highly collaborative. So in terms of products, what we did was to publish more than 100 publications. We generated 10 among platforms and big data in terms of big data omics datasets. Then we have around 20 hits for drugs. We have 8 patents filed or in filing and 1 or 2 startups. Now I will go into the detail of some of them. So here you see our 3 platforms. So the first is a single molecule photon resolved microscopy that can identify in vivo the RNA, being the RNA the target of the drug or the drug itself. Then we have a platform which is based on single molecule direct RNA sequencing, and this is really powerful because it's based on the Oxford nanopore technology, but the power here is that we are able not just to obtain the list of messenger RNA that are produced by cells in any conditions, but also to identify these decorations in terms of methylations, for example, that are now really an open field in the RNA biology. Finally, we also have another important key platform that has been mentioned before, which is a platform for the production of mRNA, which is also has a patent and is now a startup. Then we have, this is the summary of the patent in terms of disease and in terms of therapies that we have developed. Finally, this is one of the 2 startups that are the output of our spoke. And so basically this is our tag therapeutics, which is really based on, 2 patents and on targeting the RNA telomerase, which is really new in the field. And so with this, go seicho arigatou gozaimashita. Thank you for your attention. And if you want to contact us, please send me an email or visit our website. Thank you again.
Thank you very much. Thank you. Now we have Professor Manuela Elmer-Citteric, who is a professor of molecular biology and bioinformatics at the University of Rome, Tor Vergata, and principal investigator in Spoke 7.
Hi, I'm very excited to be here. I want just to say thanks to the organizers, to the local organizers, and to the hosting country. So I will talk about RNA as representative somehow of Spoke number 7, which is about biocomputing. These are the institutions involved. The leader is in Bari, Professor Pesole. Then we have the University of Milan, University of Naples, Federico II, the CNR, IIT, the University of Roma Tor Vergata, where I work, and also the University of Padova as well. So the entire spoke, I remind you, is a horizontal spoke. It's organized into 4 work packages. There's no time to go into all of them. I chose just to talk about the first one, which is very horizontal work package, because we develop methods which can be used from all other spokes. And in this case, I will concentrate just on the development of tools for RNA analysis. The 4th work package is about integrative methods for the analysis of omics data. And I have chosen autoimmune diseases as a focus case. So about the development of RNA tools. We have developed an alphabet which can help describing not only the sequence of an RNA, which is very common being a bioinformatician, but also describing the secondary structure. And also this alphabet can have inside it the possibility to include evolutionary information. So what we have done, we have built a server whose name is Beagle. So one RNA molecule sequence or many can be compared to several transcriptomes, also folded according to experimental data such as IC shape data. So coming with one molecule, the server can give you in very short time the results of a comparison. And what's the meaning of this? Is that if you have one molecule, you don't have the function, you can discover that there are similar RNAs. Maybe this can help identifying the function of yours. Then with Brio, we have taken all results of available ClipSec experiment. This means that in this experiment, individual proteins are used as baits to select many RNA molecules, and we take those molecules, analyze them in search for sequence or structure motifs. So for each one of the proteins used in these experiments, we have a certain number of statistically significant sequence and structure motifs, and we put these motifs into the database there. So the server can be used to query with one molecule of RNA that is automatically folded by the server, which ones are the protein possibly binding the RNA molecule of my interest. The third and last about the development of tools for RNA is a method to infer the interaction probability between RNA and proteins. As you know, there are already several methods. We try to build a model based on artificial intelligence, and there are several ways to use artificial intelligence. We used these large language models. For sure, everybody knows about the ChatGPT, about Gemini. It's the same, but is focused on the use of RNA sequences and protein sequences, which is it has a sense. Since these molecules are composed of letters, nucleotides, and amino acids, they seem to be words, so they can be analyzed with these models. But this is not only this. We used also a mechanism of cross-attention, which is a mechanism quite new in artificial intelligence, which helps focusing on specific sequences and places in the molecule which are being analyzed to identify better the connection. And these are the results. We have been able to compare our method, which is the one in yellow, with the two other state-of-the-art methods which have been trained on the same data set. And now I'll move to the other topic, which is about this focus. We are analyzing cases of where we can exploit methods of integrative methods to the analysis of omics data. And we decided to squeeze from GEO, which is a very well-known, very rich database of gene expression, because it's so rich, but unfortunately so unstructured, so not well annotated, let's say this, which is a real pity, because inside there are diamonds, it's gold, but how to keep it out? So we worked manually to extract from the database about 13,000 transcriptomic profiles, selecting as a test case autoimmune diseases, since this topic is so important in the industrial world, so they are so diffused. And since just analyzing gene expression is complicated with having 13,000 transcriptomes, we decided to try to go more close to the biological meaning of what we were looking at. And we selected higher order features, such as the ones that you see there. That is the cell type involved, so we used cell type deconvolution, pathway activity, transcription factor activities, microRNAs, and so on and so forth. These features are very many, as you can see. there are many of these features. Not all of them are significant for the topic we chose. So we have selected the ones between these features which demonstrated to be statistically significant in at least one of the autoimmune disease that we analyzed. And we have 700 features, which is a lot. And we decided to cluster them. And this is the clustering, according to Spearman, that we have done of these 700 features. And then we have looked at these patterns, which represent patterns of disorder in autoimmune diseases. And I'm just focusing on two of them, interferon response and inflammation, which I heard already twice, 3 times in preceding talks. The point is, okay, we have looked at these cases, at these modules, but are they meaningful? What do they mean? Can they have a biological importance? And I'll show you 3 examples. First of all, what I'm showing here is a graph, but I'll show you 3 other graphs like this. So on the x-axis, you see the samples. On the y-axis, you see the score. We have calculated an aggregated score averaging the numbers of the different cases analyzed. So these are hundreds, a couple of hundreds, if I remember well, is an independent cohort. It's not part of our cases already analyzed, of healthy people, treated and not treated with interferon. And the module is able to distinguish these cases, at 0 time, after 24 hours, 48 hours, at 0 time they are very similar, and later they are very easily distinguished and statistically significantly distinguished. So this is only validation on perturbation data. Then we went on, and again, using an independent cohort of patients, we analyzed the psoriatic lesions. And you see in the graph again, in orange, the analysis of tissue from non-lesional skin. In red, psoriasis skin lesion. And in green, the same lesion, but after treatment with an anti-inflammatory drug. So also in this case, we can distinguish the cases. And this is more complicated than the preceding case because this is on clinical data. And the last and most interestingly, I show you that this module, and specifically this module on inflammation, which seemed to be particularly rich on information, can help distinguishing responders from non-responders. The difference you see are not between treated and not treated, are before the treatment. And the data you're seeing are about Crohn's disease in the middle, and I finished, and ulcerative colitis. So also these modules have a predictive ability. And this is the group. I show you also collaboration with with SPOC 5, Professor Novelli and collaborators, and SPOC 3, Alessandro Palma and collaborators. I thank you for your attention.
Thank you. Thank you very much. Next, we welcome our representative from SPOC 9, Professor Marisa Brini, who is a professor of biochemistry at the University of Padua and principal investigator in SPOC 9.
Okay, so good afternoon to everyone, and thank you, all of you, to be here to listen our competence and to host us. So today I have the privilege to represent SPOC 9, which is an horizontal SPOC, and so it is not focused on a specific disease, but it's rather focused on developing platform ranging from target to therapy, as you can see from the title, and specifically focus on the pharmacology and on developing regulatory competence and platform for assess the delivery of nucleic acid drugs and assess the toxicity and distribution in a regulatory frame. So The spoke is coordinated by Professor Paolo Ciana and University of Milan, and as you can see from this slide, is of course spread around across Italy. We have, in addition to the University of Milan, 11 affiliate institutions among University of Milan, of course, Humanitas, and University of Pavia, University of Padova, Naples, and also Rome, University of La Sapienza, and as well Center of National Research. And of course, we have a lot of different partner, biotechnologies and pharmaceutical industry across the Italy. And altogether, we share interdisciplinary and transsectoral expertise, and with the purpose to create a competence center on RNA drug pharmacology. And we are organized our activity in 4 major work package. The first one is mainly focused on RNA drug pharmacokinetics by developing bioanalytical tools to understand the distribution, the metabolism, the secretion of RNA-based drugs. And the work package 2 is mainly focused on pharmacodynamics, especially on developing reporter system, both in vitro and in vivo models to follow RNA drug activity and the delivery of RNA drug molecules. And then, as being a pharmacological spoke, we also have a package mainly focused on assess toxicology and immunoreactivity of RNA drugs. And again, with the aim to establish technological platform to assess or to improve already existing platform to assess this particular aspect. And the last work package 4, is mainly based on developing in new regulatory strategies that are needed to use and to speed the development, the application of the use of this specific drug. And today I will mainly focus on this work package, and particularly on the work that we are doing in Padova on the developing of a specific reporter system to monitor the delivery and the efficacy of RNA molecules, both in canonical cell culture, then in 3D cell culture, and as well in vivo. So here is the group that is working on this specific topic, and I'm the coordinator of this group together with Professor Tito Cali, and some people that you see in the list are all being recruited thanks to the founding of the RNA and Gene Therapy Center. And so which is the major question that we want to address is to try to develop a platform and based on new technology that can address some important aspects. Among them, one of the major challenges is to the delivery of RNA-specific drug to specific target cells, and we realized that the way that you have to assess that the specificity of the delivery and the efficacy of delivery is that are actually available are not so easy and so quantitative to compare the different delivery system. And so we thought to develop a new probe, new family of reporter, and we decided to use and exploit this technology, which is based on this green fluorescent protein, which is a split green fluorescent protein, meaning that the backbone of the original GFP, green fluorescent protein, has been split into portions. You see here this beta barrel and this beta strand that has been removed from the beta barrel, and the two pieces are not fluorescent anymore, but they can easily recomplement or reconstitute the fluorescent signal when they came close enough. So with this tool in our hand, we decided to develop a family actually of delivery sensor, specifically considering to tag this portion in the form, of course, of mRNA to mRNA of interest, and then package them in different delivery system, and then transfer to hosting cell line or also hosting animal that express the other portion, the big portion of the protein. In this way, we should be able to follow the delivery, and not only to follow the delivery by different system, but also to assess the efficacy of the translation of mRNA of interest, because it's tagged with this small portion that is able to complement the fluorescence signal. So having this in mind, we decided to test the system by selecting as mRNA of interest, mRNA coding for a red fluorescent protein that we tag with this small beta strand and introduce in the cells, and following the increase of the fluorescent signal. And we did this in 2D cell line of different embryological origin, as well in 3D spheroids or organoid generated from iPSC cells of different, and then eventually developing different cells of different embryological origins. And here you can see our results. Here we challenge our system with increasing doses, increases amount of mRNA tag with the beta strand, and we follow the increasing of the fluorescence signal according the image that we took to automatize confocal microscope, and you see that the system is nicely working, both if we provide the beta 11 strand tagged with the mRNA of interest that is expressing a protein, in this case the red FP protein, but even if we just tag these small pieces, which is only 16 amino acid. This was done in 2D cell culture, but the system is working also in, and here we can, just quantify the signal by monitoring the fluorescence. And then here I just show you that the system is also working in a 3D structure. Here we have 3D spheroids generated from stable clone expressing the big portion of the GFP 1 to 10, and we delivered the mRNA by lipid nanoparticle, and you can clearly appreciate the signal that is increasing. And then at the end, just to conclude, I just want to tell you that we have already generated mice, a Creflox GFP 1 to 10 transgenic mice, and the idea is to generate strain mice that express the GFP 1 to 10 backbone ubiquitously or in a tissue-specific manner, and then to follow the delivery of mRNA of interest and also to quantify its expression by monitoring the fluorescence signal, both in a mice that is expressing the ubiquitous GFP 1 to 10, and in this way we can also eventually discriminate which is the precise target of our mRNA molecule, or more specifically by looking at the mice that are tissue specific, expressing the GFP 1 to 10 moiety. And with this idea, we have our future perspectives to develop a spin-off and to offer service for assess RNA delivery by rapid and efficient high-throughput technology, and eventually interrogate efficient delivery, stability, and translation of selected target RNA, and then also to move to test RNA delivery efficiency in vivo. With this, I've finished. I just want to thank you for your attention, and of course, if you need further information, want to discuss and to have more detail about this split sensor, but other topic related to the spoke, just contact me or Professor Paolo Ciana, who is the leader of Spoke 9. Thank you.
Thank you, Professor. Now joining us is Dr. Marco Silvestri, who is the project manager and medical AI lead from Isinnova, which is an industrial partner of the National Center, collaborating with Spoke 8.
Good afternoon, everyone. It's a big pleasure to be here, and thank you very much for the opportunity. As I told you, it's a big pleasure to be in such a great context like as Expo 2025. And as a head of the DIANA project, I'm here today to tell you something about the results that we are going to obtain about our project, DIANA platform, defined as a new frontier in personalized medicine to enhance clinical testing and monitor therapy outcomes. What is DIANA? DIANA is defined as diagnostic intelligence for advanced non-invasive analysis, is an advanced diagnostic platform equipped with different medical devices to deliver comprehensive health assessment. It enables simultaneous detection of several physiological parameters in a non-invasive ways in less than 20 minutes, provide an automated tool for clinical patients evaluation, usable different healthcare settings, and optimize clinical practice by reducing the cost and the time frames. When we talk about DIANA, we have to think about the DIANA role in a national center, and in particular, everything starts from Spoke 8, defined as platform for RNA and DNA delivery, and in particular, thanks to Untied Vision, that is one of the main partners of the DIANA project. The main activities of Spoke 8 are related to the development of next generation drug delivery systems, and to accelerate for the acceleration of translational pathways. And considering DIANA, we're starting from the Spoke 8 to change the paradigm. So from drug production to RNA drug therapy patient monitoring. Indeed, DIANA, thanks to the key points that I will show you in the next slide, allows to the cross-spoke activities, in particular with all the spokes, and in particular with the Spoke 4 and the Spoke 10. With the Spoke 4, I mean metabolic and cardiovascular disease, and the Spoke 10, preclinical development. But when we talk about DIANA and we talk about precision medicine, President Rizzuto already defined the concept of precision medicine. In literature, there are more than 16,000 scientific publications talking about the role of precision medicine. And during the last 20 years, this concept explodes. And in particular, if you think about precision medicine and if you think about a device, a platform like DIANA, we have the possibility to improve, to enhance the health monitoring as a tool for precision medicine. And as I told you before, these are the key points of DIANA. DIANA is a platform that allows predictive AI models and a proprietary solution based on machine learning technique and deep learning technique. Is working in compliance with regulatory for a secure data archiving following the GDPR. Is a medical grade technology. Indeed, the concept of DIANA is to collect all the medical devices, certified medical devices, to perform clinical analysis that guarantee accuracy and reliability. And at the end is rapid, non-invasive test. And as I told you before, is composed by different key points. Diana is able to analyze different body area, in particular 7 body areas. From top to the bottom, we can check brain status, cardiac status, hemodynamic status, blood composition, body composition, pulmonary status, and muscle status, using different medical devices. So, for example, electrocardiograph and a digital stethoscope to evaluate the cardiac status, to 3D body scanner and bioelectrical impedance to evaluate the body composition. And all of this technology allowed us to obtain more than 100 physiological parameters that are organized in an hybrid structure. Raw data for ECG, EMG trace, for example, and the structured data, the metrics. And this design support many downstream analysis, clinical interpretation, and AI-driven processing. And in this context, during this year, we published our first preliminary results, and in particular, Alessia, our PhD student, studied the role of a bimodal approach to evaluate and to detect cardiovascular diseases. And considering many data present in literature, we were able to compare bimodal approach, so ECG coupled with PCG, with unimodal approach. And what we found was that the bimodal approach, so the combination of ECG data and the PCG data, is a really good approach to increase the accuracy of our test and to detect in a better way, in this case, cardiovascular disease detection. We are currently working to obtain other preliminary results. And in particular, as you can see from the roadmap, we are really young, and we start after the 2020. And the first prototype will be ready at the end of June 2025, the second one at the end of October 2025. In 2024, we had the possibility to take part to this great center. And we are moving on. In 2026, we already plan to start the certification process in order to obtain the CE mark, and working in accordance to ISO 13485 and MDR regulatory. And when we talk about the future, this is the next step to the future that we are trying to do. These are the 4 key points that we are working on. Starting from the first point, we try to add new body area to analyze in order to increase the number of parameters and to increase our ability to check the health status of the patients, like as optical area, acoustic, skin. And our idea is to add new parameters in order to apply new statistical methods, and we use maybe cross-correlation analysis to derive many other parameters. The last 2 points are related to the data. We are trying to upgrade the database structure based on cloud and the data lake based to increase the flexibility and the scalability of our model. So raw data and the structured data, as I previously showed you. And at the end, we are trying to develop new AI algorithm. As I showed you in the previous slide, we are currently working in the cardiological field, but we would like to extend our ability to work with AI and deep learning methodology. For example, we are working with EMG data in order to study the age and maybe the sarcopeny of the patients. And this is our vision. And talking about our vision, I have to talk about our collaborations. We are really active in Lombardia, in Italy, and we think that we can do something really important also in the context of the center. And thank you very much for the attention.
Thank you, Marco. Great. Congratulations to all our speakers. I'm very proud for our time management. We have some time now for the audience who would like to ask some questions or maybe share their thoughts on what we presented. Again, please raise your hands and introduce yourself first. Yes, please.
Thank you very much. My name is Takeshi Omasa, Professor and Dean of Graduate School of Engineering, the University of Osaka. So I would like to ask about over the, today they did not mention detail in the Spoke 10. It's a preclinical or some transfer to the industries technologies to going to the industries. Also in Japan, in such kind of the function is very important for us. So then I would like to very briefly introduce the Spoke 10 function.
Thank
you very much.
I invite Professor Rizzuto. Unfortunately, the representatives of Spoke 10 couldn't make it here in person, but we had planned a small, few words by our president to introduce also Spoke 10.
Yes, thank you.
So Spoke 10 is originates from an already existing gene therapy consortium, which is based on 3 sites, and the 3 sites are Ospedale Pediatrico Bambin Gesù, which is a hospital in Rome focused on children's diseases. TIGET, Italy has a charity focused on genetic disorders that is called Teleton. And Teleton has its own institute for gene therapy. So Teleton Institute for Gene Therapy. And the Fondazione Tettamanti, which is a foundation that is based in a hospital in the north, affiliated with the University of Milan Bicocca, and the hospital of Monza. So they, even before we generated the new project, they were already collaborating and they were already establishing this consortium. And then they grouped in some other groups. One was mentioned because it's also on Spoke 1. That is Alessandra Biffi in University of Padua. One is in University of L'Aquila, another one is in the University of Naples. They are all, many of these are, and then I will go into what is in the current clinical trials that are being carried. There is, I would say that as of now, the 2 areas in which more intense and more active are the clinical trials are genetic diseases, and in particular, in part you've heard it because Spoke 1 has also some clinical trials going on in the center in Modena, but certainly TIGET is focused on rare genetic diseases of neurological sites, so like glycogenosis and lysosomal diseases and different lysosomal diseases. Now they have started, and part of the center is a trial on thalassemia. You know, thalassemia is highly prevalent. In our part is beta thalassemia. I know that in this part of the world is alpha thalassemia, but for us it's beta thalassemia. And as you know, there is now a gene therapy approach based on CRISPR-Cas knocking out of BCL11A, and they are implementing this on a large scale. So this is where they're focusing. So again, storage diseases and hemoglobinopathies, genetic hemoglobinopathies. The other, and which essentially is Bambin Gesù and Fondazione Tettamanti, is focused on CAR-T approaches, chimeric antigen T cells, and in particular, the group of Franco Locatelli has actually published in New England Journal of Medicine a very promising, very successful example of CAR-T treatment of solid tumors, and in particular, child neuroblastoma. In their application is on this, whereas Professor Biondi in Monza is mostly on leukemia, which is a classical application of CAR-T. So these are, I would say, are the ones that are more advanced in terms of clinical trials. The goal of Spoke 10 was really to make it happen on larger grounds. So many of these are the applications that are coming, but also already existing applications, with the idea that gene therapy becomes widely applicable in the country. So that so in there is also substantial infrastructure building. So the gene therapy facility is being expanded. I take the liberty of being here also to speak about the other spoke that is not here, which is Spoke 8, and that, as you know, is it's really also a very horizontal and transversal spoke, because it addresses a key point, which is delivery, and manipulating and changing and developing new tools to address the RNAs, whatever RNA, mRNA or the non-coding RNAs, to the tissue of interest. You've heard the vertical spokes, and many are related to organs that are very, very difficult to tackle and to reach. And so they are, again, just to summarize, from the technological point of view, they are mostly focused on engineering and developing lipid nanoparticles, so different formulation of lipid nanoparticles, AAV, so viral vectors, and extracellular vesicles in terms of, of course, then there are, every single group is changing. There is a lot of collaboration between vertical spokes and Spoke 8. For example, we are interested in targeting the natural immune system, so the macrophages, and we are collaborating with two groups, one in Padova and one in Naples, who are engineering the lipid nanoparticles for delivering to macrophages. But this is happening for much of the center with them. We ask a lot of them, because really, at this point, targeted delivery is one of the hurdles in developing. Spoke 8 also has its own screening methods. So they have cell on a chip. They're using in vivo models like zebrafish. They include some surgeons that have explants of human tissues that can be maintained for example, liver, for a long time. So they have their own screening methods, and certainly they do collaborate. You've heard with Spoke 9, the split GFP method. In Spoke 4, we have a platform based on organoids that we have developed, and of course, there is mutual exchange between their new tools and our capability of reporting and monitoring the outcome.
Yeah, thank you very much.
Please, professore. Vuole sedere qui se magari
Thank you for great talks. I'm Susumu Chiyama from Osaka University. I have about the question about RNA technology is quite relatively, I think, new technology and a new field of medicine. But at the same time, is there any concern about what is the the adverse effect or severe adverse event during the clinical trial? Because I think also recently EMA changed. I think very recently, they set a new guideline about ATMPs, which will be effective from July, I think. And so how such new guideline cover RNA therapy? Especially regarding also the adverse event, what is the main challenges about adverse effect? Or maybe immunogenicity, immunoreactivity is one of such a thing, but during the clinical trials, the people encounter the adverse event. And how to solve that problem is I'm curious about that.
Well, I think this is the key point that needs to be addressed now. We know that for RNA, we had an emergency, and the emergency was that mRNA was needed to generate a vaccine to get out of a dramatic pandemic. So we know that we had a period that the combination of speed in research and translational research, but also the speed in the regulatory evaluation was greatly accelerated. I mean, phase 1, phase 2, phase 3 for the COVID vaccines were overlapping. As soon as phase 1 proved to be efficacious, you move to phase 2 and you move to phase 3. Now that we are in, let's say, in a non-emergency situation, I think that we are really, and this is why our Spoke 9 has also a regulatory element, because we really need to investigate and evaluate with the regulatory agencies what is the procedure. And clearly we are in a completely different pharmacology area. We are in a pharmacology area in which molecules are very similar. They're not like small molecules that are radically different. We are looking at the same platform. And in this respect, it ought to be simplified. And we expect that the evaluation procedure will be probably faster, because many of the potential adverse effects of the chemical species will be evaluated. I mean, will be comparable for the different RNAs. At the same time, we need to really validate species-specific screening methods, because in terms of efficacy, but also in terms of side effects, a sequence, the sequence that we evaluate is a human sequence, and the system, while for many pharmacological characteristics, we can go on the mouse, but for many efficacy, pharmacological efficacy, but also side effects, we need to be in a human system. And clearly, our regulatory agencies are starting to evaluate much of the chemicals on organoids. So we already have a shift from the animal to the organoids, and I think that for RNAs, this will be even more necessary, because the animal system will cover only a small part of the analysis of the biological effect of the molecule. In terms of side effect, I think that we have, of the molecules that we have heard, aptamers and siRNAs and microRNAs, I think that they're probably going to be different in evaluation, in the sense that I would imagine that the single RNA specific non-coding RNA, so the shRNA, will probably have a simpler pharmacology, because it goes directly to one sequence, whereas the microRNAs very often pleiotropic, so the possibility of cross-reactivity is greater, and therefore I think that in this we will have to analyze in great detail in the system, in the screening system, what is the effect in non-target tissues. The second one is how to reach, and I think that in this respect, the new methods that we have, the organoids on the one side, and the in vivo part of which organs will take it up. I mean, how much, especially for targets that are expressed in all cells. If you have, like the example that I showed, MRF1 is expressed in skeletal muscle. So you're not, maybe there is not so relevant to how much the RNA is distributed within the organ, because the selectivity of the target will be the key element. But if you have a target that is ubiquitously expressed, then you really need to be selective in targeting your RNA to the organ of interest. And in this, I think that you will have to combine the organoids and the system like the one that we heard before, in which you can really see if you deliver, if you make a tail vein injection of a lipid nanoparticle plus an RNA, where it will go in and whether it goes really to the organ that you're interested. But certainly, answering simply to your question, it's a new pharmacology. And it's a new pharmacology in which we will take advantage of the fact that many of the results that we obtain can be extrapolated also to other RNAs, and that's an advantage. But in many aspects, we really have to create this pharmacology and validate it by the agencies.
Thank you. Great. If you don't have more questions right now, I would like to invite Professor Rizzuto to offer closing remarks.
Well, my closing remark is that if you as you have understood by now, this was a major effort of our country. I think it's a new enterprise that we have made, and it's also very novel. It is the first time that we asked essentially the entire research system of Italy, because you've seen that there are all the universities of Italy, to group up on a new technology and immediately meet also the industrial part. I think that Italy has good research activity in proportion to the size that we are. We are more or less the size of Japan, and I think that there are excellent, medium, as in every scientific environment. We shouldn't say that we are better than others, but we're not worse than others. So I think in terms of research, we are competitive. What we have lacked so far are two aspects. One is to really put together who is strongly collaborating on a new task. And the center does this. We identify the technology that we want to develop, we as a nation want to develop, and we grouped together high-profile scientists from all over Italy suddenly. So putting them together, which is managing this, as you can imagine, was complicated because we are talking about hundreds of research groups. But I think that now we have something. And the second part, we have the industrial part immediately with us from the very start. And again, if an area in which Italy is relatively weak is technology transfer from good science into application. And we hope that this national center is also an opportunity, since we are now all together and we are on a single focus, and we are a good match of good scientists and Italian and international industries that are part of the center. And I think that this is what we need to do now. And I am grateful. Well, I'm grateful to the colleagues who had a difficult task to summarize hundreds of different, sometimes selecting one rather than the other. And, but of course, it was already, I think, quite challenging like this for them and for you to get suddenly an exposure to this broad range of activities. So I'm thankful to them. I'm very thankful to you for coming, and I hope that this will be a seed in a potential collaboration between Italy and the effort of Italy in this aspect and Japan. Science is collaboration, science is integration, science is working together, and in a moment in which coming together doesn't appear to be the common theme in the world, I think that we ought to do it, and doing it with who is very similar to us in many aspects, like Japan, would be very important. So I hope that, I thank you for being here today. So I'm really grateful that to you for dedicating your time to us and getting to know what Italy is doing in this field. And I hope that from this, we can generate new collaborations, new interaction on the scientific part, on the application part, on the patenting part, on the startups, whatever I think we can do together would be very, very important. The goal of the National Center is to certainly to strengthen an interaction in the country, but also to create new collaborations and new interaction that probably coming from individual focal activities would be more difficult, while having a, let's call a systematic interaction with top institutions like your universities and in general your research system will be very profitable. So again, I thank Expo for giving us the opportunity to meet you, and I hope that from this we can generate a new interaction and new developments for us and for you. Thank you.
Thank you, Professor. And thanks again to each of the distinguished speakers. And thank you again to the audience for your participation. At this point, we would like to invite you for a short cocktail, which will give you and us the opportunity to continue this conversation. The cocktail takes place upstairs. So, yeah, we are very happy to welcome you. And please join us. Thank you.